Hello, good day everyone. Today we will discuss about the large-scale chromosomal changes. A reciprocal translocation demonstrated by chromosome painting is shown in the picture. A suspension of chromosomes from many cells is passed through an electronic device that sorts the chromosomes by size. DNA is extracted from individual chromosomes, denatured, bound to one of several fluorescent dyes, and then added to partly denatured chromosomes on a slide. The fluorescent DNA finds its own chromosome and binds along its length by base complementarity, thus painting it. In this example, a red and a green dye have been used to paint different chromosomes. The figure shows unpainted and painted preparations. The painted preparation shows one normal green chromosome, one normal red, and two that have exchange segments. A young couple is planning to have children. The husband knows that his grandmother had a child with Down syndrome by a second marriage. Down syndrome is a set of physical and mental disorders caused by the presence of an extra chromosome 21. No records of the birth, which occurred early in the 20th century, are available, but the couple knows of no other cases of Down syndrome in their families. The couple has heard that Down syndrome results from a rare chance mistake in egg production and therefore decide that they stand only a low chance of having such a child. They decide to have children. Their first child is unaffected, but the next conception aborts spontaneously or a miscarriage, and their second child is born with Down syndrome. Was their having a Down syndrome child a coincidence, or did a connection between the genetic makeup of the child's father and that of his grandmother lead to the both having Down syndrome children? Was the spontaneous abortion significant? What tests might be necessary to investigate this situation? The analysis of such question is the topic of this lesson, which is the chromosome mutations. So what is chromosome mutations? It is a large-scale alterations of chromosome structure or by changes in the number of copies of chromosomes in a cell. As shown in the picture, this is an affected child with Down syndrome. Also, chromosome mutations are changes in chromosome number and changes in the chromosome. Changes in chromosome number are of two basic types: changes in whole chromosome sets resulting in a condition called aberrant euploidy, and changes in parts of chromosome sets resulting in a condition called aneuploidy. Organisms with multiples of the basic chromosome set are referred to as euploid. Eukaryotes such as plants, animals, and fungi carry in their cells either one chromosome set, haploidy, or two chromosome sets, diploidy. In this species, both the haploid and the diploid states are cases of normal euploidy. Organisms that have more or fewer than the normal number of sets are aberrant euploid. Polyploids are individual organisms that have more than two chromosome sets. They can be represented by 3N or triploid or triploid, 4N tetraploid, 5N pentaploid, 6N hexaploid, and so forth. The number of chromosome sets is called the ploidy or ploidy level. An individual member of a normally diploid species that has only one chromosome set is called a monoploid, to distinguish it from an individual member of a normally haploid species. Examples of these conditions are shown in the table. Aneuploidy is the second major category of chromosomal aberrations in which a chromosome number is abnormal. An aneuploid is an individual organism whose chromosome number differs from the wild type by part of a chromosome set. Generally, the aneuploid chromosome set differs from the wild type by only one chromosome or by a small number of chromosomes. An aneuploid can have a chromosome number either greater or smaller than that of the wild type. Aneuploid nomenclature, as seen in the table, is based on the number of copies of the specific chromosome in the aneuploid state. For autosomes in diploid organisms, the aneuploid 2n plus 1 is trisomic. 2n minus 1 is monosomic. And 2n minus 2, the minus 2 represents the loss of both homologs of a chromosome, is nullisomic. In haploids, n plus 1 is disomic. Special notation is used to describe sex chromosome aneuploids because it must deal with the two different chromosomes. The notation merely lists the copies of each sex chromosome, such as XXY, XYY, XXX, or XO. The O stands for absence of a chromosome and is included to show that the single X symbol is not a typographical error. The cause of most aneuploidy is non-disjunction in the course of meiosis or maybe mitosis. Disjunction is another word for the normal segregation of homologous chromosomes or chromatids to opposite poles at meiotic or mitotic divisions. Non-disjunction is a failure of this process in which two chromosomes or chromatids incorrectly go to one pole and none to the other. Mitotic non-disjunction can occur as cells divide during development. Sections of the body will be aneuploid as a result. Meiotic non-disjunction is more commonly encountered. In this case, the products of meiosis are aneuploid, leading to the descendants in which the entire organism is aneuploid. In meiotic non-disjunction, the chromosomes may fail to disjoin at either the first or the second meiotic division. Either way, n-1 and n plus 1 gametes are produced. If an n-1 gamete is fertilized by an N gamete, a monosomic 2n-1 zygote is produced. The fusion of an n plus 1 and an n gamete yields a trisomic 2n plus 1. Changes in chromosome structure, called rearrangements, encompass several major classes of events. A chromosome segment can be lost, constituting a deletion, or doubled to form a duplication. The orientation of a segment within the chromosome can be reversed, constituting an inversion, or a segment can be moved to a different chromosome, constituting a translocation. DNA breakage is a major cause of each of these events. Both DNA strands must break at two different locations, followed by a rejoining of the broken ends to produce a new chromosomal arrangement. Chromosomal rearrangements by breakage can be induced artificially by using ionizing radiation. This kind of radiation, particularly X-rays and gamma rays, is highly energetic and causes numerous double-stranded breaks in DNA. So here we have the chromosome structure showing the P arm or the short arm and the Q arm or the long arm. So during the chromosomal aberrations or mutations, deletion, duplication, translocation, and all other aberrations may occur. We also have the different structures of the chromosome based on the positions of the centromere or the length of the arms. So we have here the metacentric, the submetacentric, acrocentric, in which one arm is short and the other is longer than the other, and the telocentric, in which the centromere is found at the tip of the chromosome. To understand how chromosomal rearrangements are produced by breakage, several points should be kept in mind. 1. Each chromosome is a single double-stranded DNA molecule. 2. The first event in the production of chromosomal rearrangement is the generation of two or more double-stranded breaks in the chromosome of a cell. 3. Double-stranded breaks are potentially lethal unless they are repaired. 4. Repair systems in the cell correct the double-stranded breaks by joining broken ends back together. 5. If the two ends of the same break are rejoined, the original DNA order is restored. If the ends of two different breaks are joined, however, one result is one or another type of chromosomal rearrangement. 6. The only chromosomal rearrangements that survive meiosis are those that produce DNA molecules that have one centromere and two telomeres. If a rearrangement produces a chromosome that lacks a centromere, such as a centric chromosome will not be dragged to either pole at anaphase of mitosis or meiosis, and will not be incorporated into either progeny nucleus. Therefore, acentric chromosomes are not inherited. If a rearrangement produces a chromosome with two centromeres, it will often be pulled simultaneously to opposite poles at anaphase, forming an anaphase bridge. 7. If a rearrangement duplicates or deletes a segment of a chromosome, gene balance may be affected. The larger the segment that is lost or duplicated, the more likely it is that gene imbalance will cause phenotypic abnormalities. This illustration shows the chromosomal rearrangements or the chromosomal aberrations of a cell. During deletion, one, two, or more segments of the DNA is lost or being deleted. Let us take, for example, segment 2 and 3 is being deleted. As a result, in the DNA strand, only 1 and 4 segments remain. For deletion and duplication, during crossing over, before duplication, deletion occurred in the process. So in this case, segment 4 attaches to this point. Thus, the result, we have 1, 2, 4 segment. On the other hand, segment 3 attaches to this segment and resulted to 1, 2, 3, 3, 4 segment of DNA. In the inversion, from the word itself, invert, which means that a segment, let us take for example 2 and 3 inverted. As a result, 1, 3, 2, 4 segment remain. For the translocation, which means there is a transfer of segment or a long segment of broken ends of the DNA. So let us say, for example, 1, 2 segments attached to the 8, 9, and 10, and this is the result. Whereas the 5, 6, 7 attached to 3 and 4 segment, thus forming 5, 6, 7, 3, 4 segments. And that is all for this session. Thank you and have a great day. Bye.